So I'm delighted to introduce our next speaker, Conor Giles Doran. And Conor, again, is a student of the BSc in Biomedical Health and Life Sciences. And the floor is yours, Conor. Thank you very much.
Perfect, thank you. So for my final year research project, I carried out bioinformatic analysis of copy number alteration signature mechanisms in ovarian carcinoma. And this project was carried out with Professor Donal Brennan and Dr. Bruce Moran as part of Systems Biology Ireland in UCD. So this project had the hypothesis that tumour gene expression is divergent between sets of high-grade serous ovarian cancer patient groups that are classified based on a copy number alteration signature. And the main objectives or aims of this study were to confirm CNA signature findings from a previously published ovarian cancer study, use RNA sequencing data to generate differential expression profiles for these signatures, computationally derive RNA signatures based on these results, and ultimately assess the prognostic potential of both the CNA and RNA signature classifications. So just for context, high-grade serous ovarian cancer is the most prevalent form of ovarian cancer, affecting the serous epithelial layer of the ovaries. It's associated with a very poor prognosis, causing 70 to 80% of ovarian cancer deaths. It's actually the fourth leading cause of death in women. Treatments are actually quite limited, and resistance can develop in 80 to 90% of patients who are diagnosed with widespread disease. And it involves a high level of chromosomal instability that's characterized by DNA copy number alterations. And these are amplifications or deletions of specific DNA segments within the human genome. And many studies have shown that there's a high correlation between these alterations and differential gene expression. So on the right here, you can see a simplified version of a copy number profile for a specific patient. And here each column represents a specific chromosome, so chromosome 1 to 23. And on the left is the copy number state of that chromosome, where 2 would be the normal or diploid state. And with these blue dots here, that represents the copy number state of specific DNA segments within the human genome. And if these blue dots are above or below 2, that represents a copy number alteration, so an amplification or deletion. And you can see straight away from these patient samples, there's a huge level of variability or heterogeneity across ovarian cancer, which makes it very difficult to stratify patients for specific treatment regimens. You know, this patient's profile is very different to the next patient's and so on. And that's where this new stratification method comes into play, called copy number alteration signatures. And what you're aiming to do with these is group patients based on their molecular profile. So you're trying to group patients based off this copy number alteration profile and identify patterns among the variability that's there, and hopefully match treatments to the individual based on this profile. So in my project, there were 3 components to the methods: CNA signature identification, differential gene expression analysis, and RNA signature and survival analysis. And to just break down the sampling involved in this study, I started off with 587 high-grade serous ovarian cancer samples from the TCGA, which is the Cancer Genome Atlas, which is a publicly available online genomics database. And 415 of these samples had copy number data available that was used for CNA signature identification. 311 samples were matching across copy number, clinical, and RNA-seq count data, and they were used for parts 2 and 3 of the methods. So for the first component of the methods involving CNA signature identification, as I mentioned, 415 high-grade serous ovarian cancer samples were used that had copy number data available. And the published methods of a 2018 landmark ovarian cancer study involving CNA signatures were used by McIntyre et al. And these were all carried out using the OR programming language. And this section mainly poses a learning exercise for me. I had to break down their OR scripts, understand how they operate, understand the functionalities of OR, and actually relate, you know, the fundamental code that I was writing to the biology that I was investigating. And ultimately, then I wanted to derive the signatures using the data I had. So just to give an overview of these published methods, you start off with the patient data, so the copy number data, those profiles I was mentioning, and then you compute the genome-wide distribution of 6 fundamental copy number features that were previously identified as hallmarks for specific genomic aberrations. And from there, you have to compute the optimum number of feature components for statistical models. And with a variety of statistical methods, then you can extrapolate the signatures. And what this looks like are these matrices here. So these are the results for the CNA signature identification. You end up with a normalized signature by sample matrix, where on the left is the matrix for the 415 samples, and on the right, the 311 matching samples that were used for downstream analysis. And what you can see is that 7 signatures were derived in total, with each signature containing a probability value of being associated with a particular sample here on the right. And some samples you can see are clearly defined by one signature, whereas others are comprised of a number of different signatures. And if you see up the top left as well, certain samples have a bit of a blurred boundary between signatures or a kind of an overlap. And that highlights the difficulty in making these signatures and making them precise and having unique associations with a particular sample. But the heterogeneity among samples is clearly visible across these matrices. So for the second part, I carried out differential gene expression analysis to see what genes and what mechanisms were characterizing each signature. So to do this, I used the 311 matching samples across CNA, clinical, and RNA-seq count data. So first off, I had to identify the maximum signature for each sample. However, there was no maximum signature, 7 samples, so I was left with 6 signatures in total. Differential expression analysis was then carried out with the R programming language using 3 packages known as DESeq2, EdgeOR, and Limma, of which I found the overlap to get the most conservative result. And what I was doing here was trying to find the statistical significance of a gene between 2 different conditions. So the conditions I were investigating were the signatures themselves. So I was comparing signature 1 versus signature 2 and signature 1 versus 3 and so on to see what genes were expressed. So what I resulted was differentially expressed gene sets for 15 comparisons. However, Limma results didn't really perform too well. So the overlap between DESeq2 and EdgeOR, those results were used. And then these gene sets for the 15 comparisons were used for fast gene set enrichment analysis, whereby a normalized enrichment score was generated for 40 hallmark pathways identified by the Broad Institute database, whereby this score would represent the degree of absolute enrichment of these specific hallmark pathways within my gene sets. So using these profiles, these differential expression results, I wanted to derive RNA expression signatures and assess their prognostic potential, along with the CNA signatures. So to do this, I needed to carry out single sample gene set enrichment analysis, or SSGSEA. So using the FGSEA results from the previous section, I identified the maximum signature per pathway, which allowed me to derive unique gene sets. So I ended up with gene sets per signature per significant hallmark pathway involved. And using SSGSEA methods then, I was able to derive RNA signatures for each sample involved, and then assess the survival potential of both the CNA and RNA signatures. So here are the RNA signature results. This is the data that was used to generate the RNA signatures. You can see that this is the maximum signature involvement per hallmark pathway. So on the left here are the hallmark pathways, the 40 hallmark pathways, and on the bottom you can see the 4 CNA signatures that were used to generate the RNA signatures. So there's only 4 here because numbers 5 and 6 unfortunately fell out of analysis because they weren't maximally enriched in any of the pathways. But if I take signature 1, for example, so CNA signature 1, you can see that it was maximally enriched, meaning it had the highest number of genes enriched for this specific pathway. So the angiogenesis pathway, apical junction pathway, coagulation pathway, and so on. And you can see that CNA signatures 2 and 4 are clearly dominated here, being enriched in the highest number of pathways and involving the highest number of genes. So it was these results that allowed me to get those unique gene sets for the CNA signatures, which then allowed me to generate RNA signatures, whereby each RNA signature was associated with a particular sample, representing the degree of absolute enrichment of these pathways and genes within each specific sample. And you can see again that blurred boundary is present, highlighting the difficulty in making those unique associations. But it showed that these CNA signatures could be further classified by RNA expression. So in survival analysis then, you can see on the left is the Kaplan-Meier curve for maximum CNA signatures, whereby you can see the significant results of signature 5 associated with the poor survival outcome and signature 6 with the best overall survival outcome. On the right is the Kaplan-Meier curve for the maximum RNA signatures, R2 and R4, as they significantly dominated. Unfortunately, these results were non-significant, but you can see that R2 was associated with the best survival outcome of the two. So overall, in conclusion, I was able to confirm McIntyre et al. Signatures. However, there were difficulties in reproducing their methods, and I had to use their results and data to generate the signatures. However, it was clear the high-grade serous ovarian cancer comprises a complex genomic profile involving a multitude of biological mechanisms. And it was clear that copy number does affect differential expression, as CNA signatures did represent actual, biological genes and pathways that were able to be further classified by RNA. However, there were inherent limitations to my studies. Samples were overlapping, some signatures were masked with more dominant signatures, and the heterogeneity involved made it very difficult to find unique associations. However, I do believe it reflects the nature of copy number, as it affects individual patients differently, and there's not a standardized pattern of disruption, and that's what makes it so difficult to treat. But overall, I think I can fail to reject the hypothesis, my initial hypothesis. And in the future, I'd love to see these methods being applied to similar cancers, such as uterine serous carcinoma, which has a very similar genomic makeup to high-grade serous ovarian cancer. So thanks very much for listening, and these are just the references, and I'm happy to answer any questions there you have.
Wow, that's fantastic. Thank you very much. I will stop